Okay. So, Dr. Hinkle, good morning.
Morning.
What guidelines are used to assess projects that could potentially be used as a bioweapon?
Well, how dangerous the product is, how easily it can be dispersed, if there are any treatments for it, but especially if it causes damage or can get into mammalian cells and cause damage. That's when we start reassessing why we're doing it or how to do it properly.
And if a project is considered a potential bioweapon, what extra precautions are put in place, specifically in the following areas, such as personnel in the lab?
If we had something that was potentially harmful to not only mammalian cells or catastrophic environmental bioweapon, for example, for agriculture, we would first of all not do it or neuter it in some way. Many times with toxins that are very deadly, we'll cut them into two parts and only work with one of the parts at a time, so it can't do its job out in the environment or in the cells. But in the most part, we molecularly neuter it, cut it in half or in parts to look at one function or the other. We'll usually work in a HEPA filtered VSL2 lab, laminar flow hood, use personal protective equipment such as lab coats, goggles, respirators if need be, and also gloves. But if we're at the respirator stage, we are not going to be using any of that stuff here. If we're using respirators, it would be in a BSL3 or biosafety level 3 with negative pressure or higher, and we won't be doing that here, ever.
So then what about in the general public and environment in that same case?
Well, I honestly believe that, and the CDC agrees with me, that it starts in the lab. Control of the organism or part of an organism, even DNA, should stay in the lab. We clean down everything in the lab, make sure that people aren't walking out with vials of it. There's accountability for how many tubes of DNA if we're working with a potential lethal either toxin or substrate or enzyme or bacteria or virus. We'll have logs of everything and check logs daily to make sure we have all the vials of that, and if any are autoclaved or sterilized, and that's the only way they get out of the lab, we make a note of it, just like they do in biodefense labs all over the country. You have to have a log of it, and everything is locked away, not only behind locked doors from the general public, but also locked away in the minus 80. So the minus 80 refrigerator actually can be locked at that point. Again, I would like to reiterate that we wouldn't be doing any of that stuff here.
So it seems pretty specific and pretty safe.
Yeah, for the most part, yeah. I mean, there's sometimes I'm sure there are unintended consequences, but even if there was a potential for, say, an overexpressed gene to cause damage out in the environment, we can put in what's called a suicide vector in there. So unless it has a certain resource that we can provide on the plate that's unusual, it'll survive. The bacteria or whatever is producing it will survive on the plate, but as soon as you take it away, the bacteria can't survive, so it'll die. So that's one of the safeguards, and we have plasmids that are DNA that has that backbone on it for a suicide vector, and anything that could be potentially damaging to the environment or the people, or animals, what have you. The suicide vectors are very commonly used for that purpose.
So I guess this will be a good follow-up to the last question then. Um, what are some ways to prevent the potential for the possible release into the environment for these biological elements, either by accident or on purpose?
Well, I would be remiss if I didn't reiterate the basic rule in the lab is clean up after yourself and use personal protective equipment. All my students are at least trained to the point where they have basic biosafety knowledge, good aseptic technique, which means they're not carrying around and transmitting bacteria or DNA to places they shouldn't be. Our DNA work is done in a very specific area of the room. Everything's cleaned up afterwards, and they are encouraged to use lab coats at this point. If we were using anything that was really potentially harmful, that would be enforced strictly. So if it's curtailed in the lab, there is much less likely, it's much less likely that we would have problems in the environment. The suicide vector, as I mentioned before, is important, and if any of these things did not have a ready treatment, such as an easily obtained antibiotic, or we don't have a vaccine against it or anything, we would not be using it at all. And that's already asking quite a bit, because we won't be working with anything that has an infectious disease component. The toxins that we work with are not really toxins for mammalian cells, and the environmental impact, all the things, the genes we're working with are already in the environment, in common soil organisms, so it can't really be used as an agricultural bioweapon, and they're not going into any strains that are currently agricultural pests, so they wouldn't be transmitted that way. So we looked ahead. We're making sure that nothing can potentially be spread, and that since it's commonly occurring, that's one of the curtails to making sure it's not going to harm the environment. It's already out there. It's not something that can be remade as a toxin.
All right, thank you.
Thanks.